So as John said, my paper today is titled A Stereoscopic System for Viewing the Temporal Evolution of Brain Activity Clusters in Response to Linguistic Stimuli. And it's really a collaboration between me and a postdoc in the Creative Coding Lab within the School of Information, and also with a researcher in Dr. Elena Plante's lab in the Department of Speech, Language, and Hearing Science, both at University of Arizona. So today, I'm going to present a prototype of our stereoscopic neuroimaging tool. It's ongoing work titled, the tool is called the 3D Brain+Time view. I'll discuss what motivated us to create the tool in the first place, describe some of our design decisions, give a brief overview of the mostly positive responses to the application by the domain experts in the speech, language, and hearing department, and discuss some future directions and hopefully get some feedback from you. So the 3D Brain+Time view is a specialized volume visualization application that displays clusters of statistical data gathered from fMRI machines. And the data is related to language acquisition tasks, which we'll talk about in a second. It utilizes stereoscopic visualization to enable domain experts to more effectively explore data from various vantage points. And it makes it easy for domain experts to view the temporal evolution of these brain clusters. Of the brain activity, rather. So just some quick screenshots of the application. This is obviously in 2D. You can see some brain activity on the outside of the brain. And then here's a prototype using our stereoscopic video wall. It's a passive projection system using polarized lenses. And the user can rotate the brain, zoom into the brain, turn certain clusters on or off in order to get some insight into the activity of the brain. So just a little background about the Speech, Language, and Hearing Lab. Dr. Plante's current research is focused on understanding what happens to the brain during the process of learning. And in particular, she explores, as I said, language acquisition, with the hopes that by understanding language acquisition better, potentially she or the researchers could help people with language disorders. And she's previously known for doing studies that show that there are both structural and activation differences between normal language learners and those with neurogenic communication disorders. Her research uses magnetic resonance imaging, or MRI machines, and in their lab they have one, and they force lots of undergraduates to go into it so they can study it. And I actually wasn't aware of this, and I'm not sure how aware you are of neuroimaging. So there's a difference between MRI and fMRI. The same machine allows you to do both, but MRI studies brain anatomy, and fMRI, or functional MRI, studies brain function. And this is a slide they used to explain it to me. So here's the anatomy of Homer Simpson using an MRI scan, and here's an fMRI, which studies the brain function of Homer Simpson. And somewhere in there, allegedly, is language acquisition or language activity. So MRI machines, or rather the MRI scan, is much higher resolution, but it operates much more slowly. So you normally get a single volume after a long span of time. Whereas fMRI takes lots of scans with lower resolution, but allows you to see where blood is flowing through the brain in response to particular tasks. So the goal of Elena Plante's experiments was to use fMRI to find significant clusters of brain activity related to language, and to measure how clusters appear and change over time during language learning tasks in order to generate insight as to what a normal brain does when it encounters new language, and hopefully provide information for maybe how to help or what areas to target in people who don't have normal language acquisition activity. So the study is pretty interesting, and I thought I'd just briefly describe it to you. So previous studies have shown that both adults and children can segment a sentence into individual words while listening to speech. And so the idea there is that as I'm speaking, there's actually not a pause between every word. If you looked at an audio signal, you wouldn't see any pauses between words. And yet, as English speakers, it's very easy for us, even when someone's talking quickly, to distinguish where a word ends and a word begins, and to not get confused by syllables that are part of the same word. So, the studies have shown that the way we do this is our brains automatically recognize high transitional probability coinciding with sequential syllables that make up individual words, and a low transitional probability marks boundaries between words. And here's an example of the difference between the two. If you have a sentence, it's not the robber stole money, it's the robber stole money from the bank, and yet we're able to realize that robber, those two syllables, are likely to go together and form a single word, where our E from, even though they run together, doesn't occur in very many words, or if in any words, and therefore we interpret that as a word break. And there's another example below. So the transitional probability of syllables is used during the initial learning process to segment speech streams using statistical learning. And the interesting thing about this study is that most previous studies have done neuroimaging on the brain after subjects were exposed to stimuli to study the outcome of a learning activity. And the difference here is that she's interested in looking at the learning process during the learning activity. And so I thought this was very interesting, is that humans begin learning language, both adults and children, even when they're not consciously aware of it, simply by being exposed to hearing grammatically sensible sentences. And our brains are able to quickly make sense out of sentences with the high predictability stimuli. And so the specific experiment took a bunch of subjects and played them Norwegian sentences. None of them knew Norwegian ahead of time. And here's an example sentence that was played while the subject was in the MRI machine. I don't speak Norwegian, so I'm probably pronouncing it wrong, but it's Nils, Vil, Re-Ice, Til, Kina. So that's a high predictable stimuli that the syllable re and ice appear, I guess, in various sentences over and over again throughout the testing. So the human brain is able to understand that that's a word and not a boundary between words. So what they did is they scrambled up the speech signals. And they did it in such a way that if you asked the subject if the sentence sounded scrambled, they wouldn't know because they didn't know Norwegian. So here's an example where we take that same syllable cluster, re-ice, in the one word, and they just move it to what's normally a single syllable. So Nils, Vil, Se, Til, Re, Kina. But the hypothesis is that by measuring the brain activity, you would notice different activation patterns on grammatical sentences or sentences that are normal Norwegian and sentences that are abnormal Norwegian, even if you don't know Norwegian. So they took 32 subjects with normal language learning abilities. 16 were exposed to the learnable sentences, and 16 were exposed to these ungrammatical, unlearnable sentences. And for each subject, there were 3 scan sessions that lasted 8 minutes each with a 5-minute break in between. And within each session, they heard interleaved stimulus, so lots of different Norwegian sentences, interspersed with a short break of something, I don't know, I can't remember what it was, but it wasn't Norwegian. It was maybe English or maybe some music. And again, the interesting thing is that humans exhibit these language activation patterns even when they don't understand the language. But subjects who were exposed to the ungrammatical Norwegian, on the other hand, did not exhibit any interesting brain activation patterns. And these are not computationally generated. These are drawn by hand by one of the researchers in her lab. So this is the unlearnable sentences. They applied some statistical analyses to see particular brain activity. And I guess they expected that in certain regions they know are associated with language, they thought those would light up. But in fact, they didn't light up. And in fact, the brain activity got less and less as the learning tasks went on, which maybe, the way they interpret that is that the brains didn't know what to do with these ungrammatical sentences, wasn't able to learn from it, and so basically gave up trying. On the other hand, if they play the grammatical Norwegian sentences, the brain, there's a clear difference. There's 5 different easily identifiable clusters, and some of them do indeed take place where they know brain activity that's related to language occurs. And more interestingly to them is that these clusters don't stay still. Over the 8-minute scans, again, they did 3 8-minute scans, the clusters move, the same clusters with the same activation patterns move around the brain, which is, they don't quite know how to interpret it, I don't think, but at least they're developing a map of how, what occurs during language learning. And an interesting thing about this is that the clusters aren't necessarily spatially contiguous. They're instead defined by these temporal patterns of activation. And I'm going to show just an example of what this means. So here we see, so the analysis that they do looks at all the fMRI voxels. So it's doing a volume scan. It's pretty low resolution. It's 100 by 100 by 100, but that still creates 1 million voxels. They look at each voxel independently and try and trace its activation pattern. And then, so here's an example with just they're tracing just 3 voxels over time that have a similar activation pattern. And then what they do is they do an ICA, independent component analysis, in order to map different activation patterns to the different voxels of the brain. And so here's a simplified example. On the top, you see these observed signals. Those voxels are lighting up in different ways with different intensities over different times. And then the ICA tries to separate out different activation patterns and can find, for example, the first one, those voxels are coming online and offline or pulsing over a certain rate of time. The second one's more like a step function. Nothing happens and all of a sudden it goes online. And here's kind of more of a linear function. Interesting thing to note is that sometimes these clusters, it'll find clusters that aren't, that do overlap, which is interesting to them because there may be a particular voxel of a brain might actually have multiple functionalities. And again, the resolution's fairly low, so it might be that having a higher resolution would find some separation between the clusters. So where the stereoscopic part comes in and where the visualization tool comes in came from a discussion with the researchers in this lab where they realized, or rather I realized, they told me that there were no existing tools that were useful for them, especially ones that visualized ICA clusters, independent component analysis clusters, as they changed over time. So I see I'm running out of time, so relatively quickly, their workflow was rather complicated, strenuous, involved a lot of manual labor. Even after the pre-processing of doing the statistical analyses, just to load the data and to look at it took a long time. And the main problem is it was hard for them to see the most important aspects of the data, the data they cared about, the spatial layout, and the temporal evolution of the clusters. So I'll skip through this, but basically it's painful for every cluster, for every file, for every subject. They have to click a bunch of buttons. And there's some screenshots of the interface, and they can view the clusters, but again, they're viewing it in 2D. They have to move through it at orthogonal angles. They usually have 3 windows open for each of the axes, and they basically find it a little bit difficult to intuit what's going on with these clusters. So they're a little frustrated, and it was even more complicated. It was exacerbated by the fact they wanted to see these things change over time. So they tried some 3D Viz software, but found it difficult to customize for their specific desires. And they hadn't actually they've tried 3D stuff, but they hadn't tried stereoscopic visualization because they wrote it off as eye candy. And they thought maybe it would be good for public outreach, but they didn't see how it would actually be good for them as domain experts. So part of our 3D Brain+Time view tool was to make the workflow easier for the neuroscientists in the speech, language, and hearing lab. The interface is pretty straightforward. You can turn clusters on and off. You can change the opacity of the main brain, and you can move a slider to watch the clusters change over time. There's a problem in 2D, is that once you have lots of clusters on at once, even if they are changing over time, it becomes difficult to distinguish them. So by displaying them stereoscopically, it's much easier to view the multiple clusters, especially when they overlap. So I'll just show you a few images quickly. So this is a researcher in that lab talking about interacting with the brain. Right now there's actually an operator off screen who's doing what he's asking him to do. So there's no real interaction in this case, in this version, besides keyboards. And here's a slightly nicer version with Phong shading, where we see 3 clusters. This is a time 1. I don't know if you can see what's going on. I should have brought a video, but you can kind of see the clusters changing over time. So even though this is relatively rudimentary, this was a quantum leap over what they had before. In one view, just by moving a slider, they can see these clusters appear and disappear and evolve. And then, of course, here's a version with opacity turned on all the way, where you can see the clusters coming on and off. And you can change the opacity so it's somewhere in between. You can see a little deeper into the brain and then rotate the brain. So in conclusion, I see I'm out of time. It's important to work closely with domain experts in order to figure out what's actually useful to them. So just doing stereoscopic, they wrote off. Just doing the temporal sliders, they didn't care about. But putting them together, they found was useful. So one wasn't really useful without the other, but once they were together, they were super excited and want us to continue working on it. So lots to do. The main one is to make it not just a visualization tool, but more of a visual analytics tool where we can actually include some of the pre-processing choices with the tool to be able to show the statistics in the visualization, not just the final clusters, to change the thresholds, to annotate the clusters, and to merge the whole thing into one tool. And finally, right now we have, as you saw, we have this on the video wall in our lab. It would be nice to buy them a low-cost 3D projector or monitor, or maybe to use some HMDs so that they can use it in their lab. And I'll take some questions. Thanks a lot.